My name is Mr. Houck, and today we're going to be playing Classroom Jeopardy.
All right, question number 1. Who was the first to study genetics?
The mitochondria is the powerhouse of the cell.
Hohmann, see me after class. So actually, genetics, the first person to study genetics was the Greek physician Hippocrates, and he had a theory that suggested that particles called pangenes travel from each part of an organism's body to the eggs or sperm, and then are passed down to the next generation. However, his hypothesis is incorrect. Question number 2. Which geneticist laid the framework for modern genetics?
Gregor Mendel?
That is correct. So actually, Gregor Mendel studied pea plants in the Abbey Garden. He was a monk who lived and worked in Abbey in Brunn, Austria. And he basically studied flower color, flower position, seed color, seed shape, pod color, and stem length. And he basically studied how those traits were passed down to the offspring of the flowers. When Mendel performed a monohybrid cross between purple flowers and white flowers, he observed that all of the offspring were purple, thus concluding that purple was the dominant allele. In the third generation, he saw that 3/4 of the plants had purple flowers and that 1/4 of the plants had white flowers. For question 3, we will be doing a Punnett square between a homozygous dominant brown dog and a heterozygous white female dog. First one done, put your paper on my desk. Egelhoff, eyes on your own paper. As we observe the results here, good job, Jacob. Jacob wins the point there, 100%. And Matthew Hohmann, you're going to have to see me after class. I don't like your answer on this paper here. Square between a homozygous dominant individual and a heterozygous individual. In the upper left square, we see parent 1 contributing a dominant allele and parent 2 also contributing a dominant allele. In the top right box, we see parent 1 contributing a dominant allele and parent 2 contributing a dominant allele, therefore making it 50% homozygous dominant. In the bottom left corner, we see parent 1 giving a dominant allele and parent 2 giving a recessive allele, therefore making a heterozygous individual. In the bottom right square, we see parent 1 contributing a dominant allele and parent 2 contributing a recessive allele, therefore making that individual heterozygous. A Punnett square does not show actual results, only predicted. For question number 4, write everything you know about incomplete dominance. Hohmann, you all right? Let's see you, Jacob.
Ouch.
Hohmann, see me after class. Chill out. Incomplete dominance is when we see a mix between the two parental phenotypes. An example of this would be if you were to cross a red flower with a white flower. All of the offspring would have pink flowers. The third phenotype results from flowers of the heterozygote having less red pigment than the red homozygotes. For question 5, tell me everything you know about codominance. Jacob.
The central dogma of biology is DNA to RNA to protein.
Yeah, sure, man. Hohmann, what do you got?
Isn't codominance when two alleles are equally expressed in the offspring generation?
That is correct. Good job, Matt.
Let's go!
For question number 5, What is the difference between X and Y chromosomes, and how do they relate to sex-linked genes?
X and Y chromosomes differ because X chromosomes are much larger than Y chromosomes. They're related to sex-linked genes because X chromosomes they both can carry either dominant or recessive traits. For Y chromosomes, that trait would only show up in males, as only males have Y sex chromosomes. Females have two X chromosomes, and males have an X and a Y chromosome.
Good job, Hohmann. For question number 6, explain the difference between pleiotropy and polygenic inheritance. Hohmann.
Deshaun Kaiser sucks.
That is incorrect. See me after class, Jacob.
Pleiotropy is when one single gene affects multiple phenotypes, while the other one is multiple genes affecting one phenotype.
Good job, man. For question number 7, what does DNA stand for, and what is it made of? Yes, Egelhoff.
Pass.
Hohmann?
DNA stands for deoxyribonucleic acid. It's made of a sugar phosphate backbone with the phosphate further and then connected to a sugar, which is then connected to a nucleotide. Those can be either A, G, C, or T.
Good job, Matt. We now have a message from our sponsors.
You could go to 5 or 6 stores or just one.
You can go to 5 or 6 stores, or you can go to one.
Well, you could go to 5 or 6 stores or just one.
You could stop at 5 or 6 stores, or just one.
You could go to 5 or 6 stores, or just one.
You could go to 5 or 6 stores, or just one.
You could go to 5 or 6 stores, or you could just go to one.
You could go to 5 or 6 stores, or just one.
You could go to 5 or 6 stores, or just one of them.
That's another Giant Eagle advantage.
Welcome back. For this question, what are the parts of the cell cycle?
Blood doping.
Jacob?
I don't know what to say. That's what you said to say.
The parts of the cell cycle are G1 phase, S phase, G2 phase, mitosis, and cytokinesis.
Good job, Hohmann.
The cell cycle is composed of 5 major steps. The first phase, G1 phase, is when the cell initially grows. The S phase is when the DNA replicates in the nucleus. The G2 phase consists of the cell preparing for division. Mitosis is when cell division occurs, meaning that there are 2 complete sets of nuclei and organelles. Cytokinesis is the final separation of the cells, when the cell wall or when the cell membrane is completely split.
For the next question, what is meiosis?
I got this.
Yes, Jacob.
Gamete formation. Meiosis consists of prophase 1, which includes homologous chromosomes composed of 2 sister chromatids coming together as pairs in a process called synapsis. Spindles will also form in prophase 1. Following prophase 1 is metaphase 1, where these homologous chromosomes line up in the middle of the cell and cross DNA over. This results in more genetic diversity. These spindles will then attach to the microtubules and pull the sister chromatids apart, which is a part of anaphase 1. Anaphase 1 is followed by telophase 1 and cytokinesis, where the sister chromatids will move to opposite sides of the cell and the cell will divide at the cleavage furrow. After cytokinesis, the 2 gametes will enter into meiosis II. Meiosis II is mostly like mitosis with the differentiation of sister chromatids.
For the next question, what is the difference between DNA replication and transcription?
Trick question. There is no difference.
Go to Ricky, babies. Get out of here. Get out of my class right now. Hohmann.
Isn't replication when DNA replicates itself into more DNA? And transcription is when DNA transforms into RNA?
That is correct. Egelhoff, I see you.
DNA replication is simply doubling the number of DNA present. This is done by making a conjugate strand of DNA. The nucleotides T and C respectively bind to A and G, making two new strands that are opposites. Transcription is a similar process. However, the conjugate strands are RNA instead of DNA. The major difference is the T nucleotide changes to U.
For this question, what is translation? Yeah, Jacob.
RNA to protein?
That's correct. Translation is when RNA is translated into an amino acid using the genetic code. A functional protein will start with AUG and end with either UAA, UGA, or UAG. For the next question, what is AIDS, what does it make DNA from, and why does Magic Johnson have it?
Isn't AIDS airline-induced divorce syndrome?
Yeah, shut up, Hohmann.
AIDS is acquired immune deficiency syndrome. It occurs when the HIV virus invades our T lymphocytes to create DNA from mRNA, and Magic Johnson acquired it from not using protection.
For the next question, what are the different types of mutations?
The different types of mutations are silent, missense, nonsense, and frameshift. In fact, the different types of mutations are as follows: substitution, deletion, and insertion. In a substitution mutation, one nucleotide is substituted for another. In a deletion mutation, one nucleotide is completely removed. In an insertion mutation, a brand new nucleotide is inserted. These different types can have varying effects and are as follows: a silent mutation, which has no change to the final codon, such as GAA to GAG, as they code for the same amino acid. A missense mutation, which changes one amino acid to another. An example would be GGC to AGC, which would change the amino acid from serine to glycine. Nonsense mutations can also occur. This is when an amino acid codon changes into a stop codon. An example would be AGA changing into UGA, which is the stop codon. Finally, a frameshift mutation changes which three nucleotides form each amino acid.